As foreshadowed by Bruce, Dr. Aaron Dunlop from the Ministry of Natural Resources is going to talk to us about next generation Lampricides and whether that's a proactive approach to sea lamprey resistance. Aaron, welcome.
Okay, so my name is Aaron Dunlop. I'm a research scientist with the Ontario Ministry of Natural Resources. Many folks may know me as someone sort of working mostly on fish population dynamics in Lake Huron, most particularly, and I do a lot of work on Lake Whitefish. But this is sort of like a little bit of my other life, working on sea lamprey and helping the Commission with its mandate for sea lamprey control. I'm giving this presentation, it's co-authored with Nadia Carmesini. Nadia was actually going to be here to give this presentation because you'll see that the next generation Lampricide part of the talk is really her baby, her work, but she was unable to get here because of the travel restrictions. Okay, so with this presentation I hope to address three questions. First of all, what is resistance? Why should we care about resistance? And then the second half of the talk is really going to focus on how can next generation Lampricides help address the risk of resistance. Okay, so I wanted to provide a definition of resistance so we're all on the same page as far as what it means. So quite simply, resistance is a genetic-based change in the susceptibility of a pest population to control measures. Resistance is a global problem in pest systems. So people have probably heard about insects developing resistance to pesticides used to control them, for example, in crop scenarios. You've probably heard about antibiotic resistance. You can also see resistance in human pests like rat populations that start to evade bait with pesticides, for example. So it's a global problem and it costs, on a global scale, a lot of money and a lot of effort. And research has been devoted to trying to mitigate the risks of resistance developing in these pest systems. But with all of these different examples, the end result is basically the same, and that is that resistance results in the reduced effectiveness of your control measure and increased pest population abundance. So much so that sometimes, using a pesticide, for example, that pesticide can no longer be used for that pest system. So this concept is, it's translatable to the sea lamprey control situation. So here what I'm going to do is just kind of walk you through a little bit of what resistance kind of looks like, I guess, with some graphs here. And I'll speak about it in the context of sea lamprey. So here we have our population. So the green part of the population, so think about this as our sea lamprey population, and all the green individuals in this population are, when they're exposed to TFM, the pesticide used to control them, they die as a normal lamprey would. So these are larval sea lamprey. However, there's a small proportion of the population that may actually survive that treatment. And in general terms, and we've heard this throughout this morning's talks as well, we generally think of the TFM being about 90% effective when it's applied to a stream. So there's always going to be a few surviving individuals. And so that's the red part of that curve. And so when we're talking about resistance, what we're talking about is these individuals have something in their genes that allows them to survive that pesticide treatment, whereas all the other individuals in the population die. So these individuals may possess resistance alleles. And these individuals can come about in the population through different means. So there could just be some genetic variation in the population. So this is based on maybe the founding population that came into the Great Lakes. There may be a few resistant individuals out there. They're just very, very rare, and so we haven't detected them potentially. The other way that resistance can arise is through novel mutations. So these are just random changes in the genes of these sea lamprey that suddenly allow them to survive that treatment. And then the other way that resistance alleles or resistant individuals can come into the population is through something like migration. So if there's still maybe a little bit of gene flow from the native range, it's possible that some genetic variation, some resistance could come in that way. So what happens is these individuals that survive the treatment, of course, they're more likely to reproduce and pass on their genes to the next generation. So these individuals have a fitness advantage in your population. And so over time, what can happen is these individuals survive, and basically you begin to see resistance emerge in your population. So this allele can actually completely take over the population because there's such a strong fitness advantage. So now we see only a small proportion of that population dies as a result of your control measure, and now we see an explosion in the pest population because the control measure is no longer working. Now it takes time for this resistance to develop, and of course we're very concerned with how long this could happen, right? And so there's a number of different factors that influence how quickly resistance could develop in a population. So this includes generation time. So, you know, obviously like a microbe or an insect has a very short generation time. So we would expect resistance in real time to develop much quicker in that kind of an organism, right? Because, it might take only months in that case. The generation time of a sea lamprey obviously is longer. Another factor that influences how long or how quickly resistance may develop is fecundity. Unfortunately here, sea lamprey have a very high fecundity. Gene flow can matter, so how much gene flow there is between different populations or populations that are more or less exposed to the pesticide. The genetic nature of resistance, so if it's a recessive trait or a dominant one, if it's the result of changes in many gene loci, for example, this is going to influence how quickly resistance could develop. As well, the selective pressure from the pesticide, so how many individuals are actually dying as a result of the pesticide. This will influence the speed of resistance development. And finally, if there's any fitness costs associated with resistance. So sometimes, if you see this random mutation arise that makes an individual resistant, well, there may be some other cost to that animal's fitness. And so the extent of those costs could really slow down resistance from developing. Okay, so there's different types of resistance that can develop in a population. So there's two here that I'm showing. The first one is behavioral or life history adaptations that limit the exposure of the lamprey to the pesticide. So these still have to be genetic-based changes, but basically, if there's a behavior out there, just like a hypothetical example, if a sea lamprey, can burrow deeper into the sediments and avoid exposure to TFM, for example. Or if the lamprey undergo metamorphosis earlier in the season and out-migrate before TFM is applied. That's an example of a life history or behavioral form of resistance. The more common one that we think of when we think of resistance is physiological resistance. So these are genetic changes that result in reduced toxicity once the pesticide enters the body of the lamprey. So it's a genetic-based change again, but it just allows that lamprey through some physiological process to survive treatment. Both of these are important because both types of resistance are going to result in reduced efficacy of the pesticide and potentially increased pest population numbers. So both need to be considered. And I've kind of just alluded to this, so why should we care about resistance development? Well, obviously, if resistance to TFM were to develop, this would increase the number of sea lamprey in the population and really threaten the sustainability of our fisheries. We've seen examples, Mike was showing some of the history of sea lamprey control and what sea lamprey, how they, really decimated lake trout populations, for example. This is what's at risk when we think about resistance. And of course, TFM is really the cornerstone of sea lamprey control program. And we want to continue to be able to use this pesticide potentially into the future. And unfortunately, if resistance were to develop, it would make TFM less effective at controlling the sea lamprey population. Okay, so I just wanted to quickly talk about, what do we know about resistance in sea lamprey thus far? So there's been a number of different research studies done over the past several years. And what these studies show are a number of things. So something that we found early on is that enough generations have passed since TFM began to be applied in the Great Lakes, and the selective pressure from TFM is high enough for resistance to develop. So, despite the fact that sea lamprey have a longer generation time, when we look to other studies where resistance has developed, or other studies where fish populations have evolved in response to some stressor or chemical in the environment, we see the time period still fits within the length of time that TFM has been applied in the Great Lakes. We have studies showing that selection from TFM treatment, this is based on modeling work, could act on specific life history traits, making control less effective. So we do need to think about this form of resistance as well. However, the really good news is that it doesn't look like full-blown physiological resistance has yet to develop in sea lamprey. So really important take-home message. And this is based on toxicity testing that's been done at Hammond Bay. So when we look at, for example, the lethal concentration required to kill, 50% of lamprey or 99% of lamprey over time, that lethal concentration hasn't changed. So this is really good news. And we haven't been hearing from agents out in the field that suddenly lamprey are surviving TFM treatment. So we know TFM is still effective and it's still working. So this is good news, and this is telling us it's not too late. We have time to think about this issue. However, there is a few red flags, I would say, in the science. One is that there's some genetic analysis which is showing there are some potential early signs of resistance developing in sea lamprey. So basically this was a study done where lamprey from the native range were exposed to TFM as well as lamprey from the Great Lakes. And what was observed was there's differences in gene expression in these different populations, and those differences occurred at places in the genome that appear important with oxidative phosphorylation, which is related to the mode of action of TFM. So this is a little bit of that red flag kind of telling us, okay, something may be happening at the genetic level in sea lamprey populations in the Great Lakes. So we need to keep our eye on this. And collectively what this work really shows is that this is something that we do need to take seriously because of the risk that it poses. Okay, so what are we doing to address the risk? Well, there was a resistance workshop held back in, I think it was 2014. And out of that workshop, there was a couple of papers that were done if folks are interested in reading more about this. But a key step that came out of that workshop was the development of the next generation Lampricide discovery project, which I'll speak about in the coming slides. As well, there was a second resistance workshop held in 2023, and out of that workshop, basically the purpose was to develop options for monitoring for resistance. So is there existing information being collected in the sea lamprey control program, or is there new information that we should be collecting that could allow us to monitor for resistance development? And of course, there's been ongoing research being conducted. Okay, so now I'm going to switch gears and talk about Nadia's work on the next generation Lampricide discovery project. So Mike Siefkes was presenting earlier about some of that initial screening that was done in the 50s and 60s when the search for Lampricide began. So during that time, over 5,000 chemicals were tested, which resulted in the discovery of TFM and niclosamide. And since that time, millions of other chemicals obviously have been developed. So there's a whole host of sort of existing information that could be mined to try to look for an alternative or a next generation pesticide that is effective against sea lamprey. And so some of the key questions being asked when looking for this chemical is, does it use a novel mode of action? Is it specific to sea lamprey? Is it cost effective? And finally, is it safe or green, so environmentally friendly, for example, and socially acceptable? And it's that first question, using a novel mode of action, that's so critical when it comes to resistance. And so why that matters is, like in other pesticide systems, we know that when you rely on a single pesticide, this really increases the chances of resistance developing. And so if we could find an alternative to either, maybe supplement with TFM or to replace TFM, this would sort of strengthen our ability to mitigate the risks of resistance from developing. As well, it would provide an alternative should resistance develop to TFM, right? We want to be able to have something to fall back on. So there's 3 stages in this project, and the first stage involves doing in silico screening. So this in silico just means computer-based screening, as well as in vitro chemical screening. So in vitro here just means developing cell lines and testing the toxicity of these cell lines to different alternate, potentially green Lampricides. The second stage is in vivo efficacy and safety testing. So here this involves doing testing of the whole organism. And then the third stage is the sort of a whole host of regulatory studies that needs to be done in order for a chemical to sort of go forward and be used. So in terms of progress, so using these in silico methods, basically what this involves is like screening existing chemical databases to look for potential chemicals that could be effective against sea lamprey. And so these kinds of, this kind of screening involves, you basically need information on the chemical structure of different chemicals, as well as information on toxicity to sea lamprey and closely related species. So you need both of these types of information together. So when the team started looking for this, unfortunately what was found was that there was not a lot of toxicity data for many chemicals specific to sea lamprey. So there just wasn't a lot of data or information out there that could be useful. So instead they had to go to the earlier studies that were done in the 1950s, and they actually went to Hammond Bay and they entered records that were existing there on these different chemicals. And through that, they discovered 16 different chemicals from 14 different classes that appeared to be selectively toxic to sea lamprey. So now they're working with those 16 chemicals to do further toxicity testing to see if there could be an alternative within those chemicals. And the next step in this process is the in vitro. So this is the development of cell lines, where now you take, so the idea is to now test those 16 different chemicals. So basically you grow these cell cultures and then you test the toxicity of these cells to those different chemicals. They tried with sea lamprey for 2.5 years to grow sea lamprey cells. Unfortunately, they were unsuccessful in doing this. And so now they've had to switch to some surrogate species, including lake sturgeon and rainbow trout. And for this work, they've been growing gill tissue or gill cells and liver cells specifically. And when they're doing this work, they're looking for toxicity with relation to 3 different functions of the cell: lysosome integrity, metabolic activity, and cell membrane integrity. So they test these 3 and how they respond to the chemical. So they've begun by, first of all, just looking at TFM and how the cell lines respond to TFM. So this is just showing some results for the gill cells for rainbow trout and lake sturgeon. And essentially what's been found so far is that, unfortunately, those gill cells, when you compare lake sturgeon and rainbow trout, there's not a lot of difference in toxicity, whereas earlier work done by Bogart et al. On the whole fish did find differences in how these fish responded. In this case, this is TFM. where lake sturgeon are more sensitive. And so what this is telling the team is that the gill cells may not be appropriate for testing these chemicals. So the next step involves looking at liver cells, which they think may offer a more viable solution for looking at this further. Okay, so basically to summarize, 14 different classes are being evaluated through these computer-based models. The fish cell lines are in development. The team is making progress on this work, important progress, but definitely more work is needed to continue along this path of trying to find a next generation lampricide, and ultimately, of course, the goal of reducing the risk of resistance. And Nadia did want me to share that some of the workforce reductions that have taken place, as well as potential future cuts to USGS, really threatens this project in the future. So with that, I'll say thank you. One minute.
Thank you, Erin.